Peptide pals, listen up. If you're missing this crucial insight, you're losing vital research time. Your chief investigative scribe, Kai Rivera, is buzzing over a breakthrough. Our immune system's T cells need a critical, secret handshake with peptide-Major Histocompatibility Complex, pMHC, to identify cells for research. But studying these interactions has been incredibly difficult because the MHC component is notoriously unstable outside a living cell. This instability has significantly hindered our understanding of T-cell recognition in various research contexts. We've been unable to fully map these unique interactions. Prepare for SMART MHCs. For ages, scientists aimed to understand T-cell recognition, but traditional MHC molecules were complex. These proteins naturally present peptides to T-cells on cell surfaces in a research context. However, isolating them for study in a test tube revealed extreme instability, like a collapsing structure. This inherent fragility made studying their fundamental mechanisms exceedingly challenging for researchers. Researchers face significant hurdles trying to analyze intricate T-cell recognition dynamics in the lab. Imagine analyzing a complex molecular dance where the key partners constantly destabilize. This instability seriously impeded understanding critical aspects of cellular interaction and recognition pathways. A common method involved growing protein parts separately, often using Escherichia coli bacteria. Then researchers painstakingly refolded them into correct 3D shapes with specific peptides. This refolding process for MHC class I heavy chain molecules was notoriously difficult and inefficient. It was time-consuming, yielded low quantities, and often felt like assembling complex items without instructions. MHC class I molecules also required helper protein beta-2 microglobulin, beta-2M, and a peptide. Without these components, the MHC molecule remained unstable, like a Jenga tower. Studying empty MHCs or easily swapping peptides became practically impossible due to this. Expressing these complex multi-part proteins in a soluble form in E. coli was another major obstacle. This significant roadblock hindered efforts to map the vast dictionary of T-cell interactions. The challenges were immense, limiting large-scale research into T-cell recognition mechanisms. All right, fellow peptide adventurers, we've seen the struggle, but now for the molecular marvels. These challenges in protein stability have been monumental, yet the solutions are here. For more insights on cutting-edge peptide news, make sure you're following me, Kai Rivera, and subscribing to Peptides today, wherever you get your research fix. We're about to explore the brilliant design that changes everything for immunology research. Let's dive in. Science heroes developed SMART MHCs de novo designed protein domains for stability. These are like building a super sleek molecular submarine from the ground up, entirely new. SMART MHCs replace wobbly parts with a custom internal scaffolding, enhancing stability. This buttresses the peptide binding groove, maintaining structure independently for research. They maintain stability without traditional B2M helper proteins or constant peptide presence. It's like the protein learned to stand on its own, with built-in structural integrity. These are produced solubly directly in E. coli, acting as efficient protein factories. This eliminates agonizing refolding steps, simplifying reagent production significantly for researchers. SMART MHCs are highly effective at binding peptides and T-cell receptors, functioning robustly. They also improve protein display on yeast cells, powerful for screening new interactions. These molecular maestros make high-quality MHC reagents accessible, a research game-changer. Having stable, easy-to-make SMART MHCs enables crucial T-cell repertoire exploration. T-cells are immune system super sleuths, identifying threats by recognizing peptide clues. Previously, only a small fraction of pMHC interactions could be effectively studied. Now the entire dictionary is accessible for high-throughput screening of interactions. This speeds identification of T-cells specific to various antigens for research purposes. It enables precise characterization of interactions, advancing immune engineering research possibilities. Honestly, these SMART MHCs are a total mic drop for immunology research. By solving MHC instability, we gain deeper understanding of T-cell biology. This accelerates research into vaccine development, immunotherapies, and clearer insights into autoimmune mechanisms. Follow me, Kai Rivera, and Peptides today for more unearthed epics. Before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment.